Mexico every year to work with us. They're studying veterinary medicine and specializing in bee health. And so they come up here to do an internship with us as well. And then we have graduate students and research collaborators that come from all over the world to help us out with our projects. So I do like to share as much credit as I can because I get to work with some great people. This is what I like to call a drone's eye view. It's kind of literally a drone's eye view, both drone camera and the drones that are flying around our site. We're in the Arboretum on the edge of the University of Guelph, and you can see some of the hives scattered around here and our facility out there. We are moving to a new location, but it's just down the road, and we'll talk about that in a little bit. So, of course, we keep bees working here in the education and research field in apiculture. We manage about 300 colonies of bees. Those provide us with the colonies we need for research projects and also to do the teaching that we do, both at the university level and at many other levels as well. We have, on an average year, up to 500 colonies involved in projects because we do collaborate with other beekeepers and with the tech transfer program. This is our home apiary, the backyard of the home apiary. At the moment, we have 150 colonies in this apiary. We just moved our bees out of our indoor overwintering room on site here. And so that's why we have so many in this location at this time. Those will be moved out as we assess them into some of our oat yards. We produce honey and all sorts of other hive products. We try to involve ourselves with every aspect of apiculture. And I feel that's really important because if we're going to teach people how to do things, I think you really should be doing them yourself. And it's interesting. So we get involved in producing all sorts of hive products. We sell about $10,000 worth of products out the front door every month. And so that also helps us keep the students and everybody employed that help us out. There's a year's production of beeswax. We divvy it up into a variety of different sizes for sale. At this point, we use the majority of the wax ourselves for candle making, lip balm, a variety of different products that we produce with beeswax. We also produce pollen. I'd like to mention, because I'm kind of proud of this fact, that the original pollen trap was developed here at the Ontario Agricultural College, and it's called the OAC pollen trap. So every other pollen trap out there is based on the design that came from here back well before my time. But we collect pollen every year, not so much to sell. I find it too time consuming to clean it and sort it. So what we're collecting it for is to use with the colonies that we raise queen bees in, because we feed them as much natural pollen as possible so we get the highest quality queens that we can. Speaking of the queen ring, this is a frame, a graft frame, and this has just come out of our cell builder, and you can see all those queen cells there. The methods we use, we try to constantly improve on them so that we get good acceptance of the queen cells and nice big cells so that we get good quality queens. We're Buckfast bee breeders. We're the only registered Buckfast breeder in North America. Buckfast bees were developed by Brother Adam, a monk at Buckfast Abbey. And he developed a system of breeding bees, and we follow that system. Part of that includes isolated mating, which we do on isolated mating islands in Lake Simcoe, up in your direction there. Brother Adam used the moors where there were very few trees around, and so not much potential for wild bees. And he had an area that was set aside that he could use so there were no other beekeepers in that area. So he got his isolation that way. We get it by moving colonies up to Georgina Island and Thora Island. On Georgina, it's owned by the Chippewas of Georgina, and we've established relationships with folks in the band there and have trained them with beekeeping. So they help us out, and have some of them have some of their own collies too with our drone lines in them. So that works out really well that we can collaborate on that. Some of the things that we're breeding for in our collies and with our queens are overwintering ability. So we have a really simple method of measure overwintering ability. If they live in the spring, they are potential breeders. If the colonies are dead, we eliminate them from our breeding program. So there's a pretty simple method. This is where it helps if people are nearby and can snicker a little bit, but anyhow, we'll move on. We have gentle bees. That's one of the things that Brother Adam was very focused on in his breeding work there. We found with our research, together with Purdue University, Dr. Guzman, my supervisor, worked with Greg Hunt, looking at defensive behavior in colonies, and we found from that research that defensive, or the flip side of that, gentle behavior, is inherited from the drones. This is a really useful fact. If we can control who our queens are mating with and make sure they're only mating with drones that come from colonies that exhibit gentle behavior, we can pass that on to the next generation. This little fellow showed up in a bee yard. We're wrapping the yard up. This is up in Thor Island. He and his dad just wandered into the bee yard, and I was sure glad that we had nice gentle bees when he showed up on site. His dad stood back and took this picture and passed it on to us. This is my boss, Dr. Ernesto Guzman, with the hat on there, the Mexican hat. Ernesto came up from Mexico 16 years ago to work with us. He's a fantastic bee researcher and a really good manager. I've never had a better boss. But this is one of our classes that we teach at the university level. We have 700 students a year take an apiculture course of some sort. And so this is people's first introduction to bees. And it happens at a time of year when robbing behavior just gets going. So it can be a bit of a challenge to have hundreds of people come through in a day on some of the tours that we do, but we make it work. But I'd like to point out that those people that you see there are the future of our industry and our profession. People learn about bees, get inspired by them, and go on to work with them. This is where I got my start in bees and beekeeping, was taking this course that Ernesto's predecessor taught. The fellow you see in the background on the left was a volunteer at that point. He is now a PhD candidate doing some really great research work. And a young woman you see on the right, also in red, the sleeveless top there, another young woman from Mexico that has gone on to do some great work in Mexico with these. But other examples of people that came through here, virtually every one of the tech transfer members who you'll often, many of you will know, started working with us, took this apiculture course and worked with us, and then have gone on to the tech transfer program, which we started back in 1990. We had a research project breeding for tracheal mite resistance. It became popular enough in the industry that we established a tech transfer program, first of its kind in North America, and now there is one in virtually every province and every state. We teach weekend-long beekeeping courses. Some of you have come down and taken these courses. This was back when we fit 24 people into a living room. We now fit 36 people into our living room. And well, pre-COVID we did. I don't know what we're going to do afterwards, but we're this year offering our introductory level beekeeping course, a weekend course. We're offering that online. If you're interested in taking that course, the registration information is up on our website. We're going digital with just about everything we can this year, and it's been a heck of a lot of change, but our course registration is done online with software called Eventbrite, and that's working out really well. So if anybody's interested, please check that out and join us May 1st and May 2nd for that course. The course content will be available for a week, so you can take it on the weekend, but you can also review information or take a break and catch some of it later. And so we're looking forward to doing that for the very first time. But we're thinking in the future that any courses we do will be more of a hybrid course, where part of it will be online and part of it will be hands-on. You just can't replace good hands-on experience.
Excuse me, Paul, you said check out the website. That's the University of Guelph website.
Yeah, if you just Google University of Guelph honeybees, you'll find our site.
Okay. Thank you.
No problem. Thank you, Bert. So we do, in normal years, we do a lot of tours. We have 5,000 people a year come to our facility for tours. We have volunteers, and those volunteers, that fellow on the left is Al. We tend to specialize in student volunteers and retired people volunteering with us to help us out. The little fellow on the right was here for a tour. Isn't he as cute as a button in that little bee suit there? Barely as tall as the hive. But we get lots of school groups coming in and some of the other groups that I've already mentioned. We have a student apiculture club. This was the 2017-2018 student club. We get about 20 students a year that join in with this club. They do help us with some bee yard activities in the fall. But the student year doesn't really overlap an awful lot with the beekeeping year. So we get them involved with some things in the fall, harvesting honey, and then they help us make products. So they're making beeswax food wraps on the left, lip balm on the right. We do all sorts of candles, package honey, and they have sales on campus once or twice a year. And the proceeds from those sales we donate to beekeeping development projects around the world. So every year they donate $2,000 to $3,000 to beekeeping development projects. And so there's a goal with the work that they're doing with our producing our bee products and selling them. This is our student apiculture club, just the executive of the club back in 1912, 1913. We think the club's been going since the 1800s, but this is the earliest record that we have. There's a number of things that we do for extension, and our website's a great place to go and explore some of these areas of work that we do to help transfer information out to beekeepers. So I won't dwell on this a lot, but I will encourage you to have a look at our website. We do YouTube videos, and I did them initially to start as a backup for the courses that we teach, because it's kind of overwhelming, all the information that people learn in a weekend on our beekeeping courses. So I started out with that goal in mind, but one of the students suggested we put them on YouTube, and that's worked out well for us. We are now over 5.4 million views of our videos, but a lot of people in other countries have kind of pirated them and put them on their own sites, and so they're now in 5 different languages, and we can't keep track of the views that are on other people's sites. I've actually encouraged that people take them, put them on their websites, translate them. It's just getting good information out to beekeepers, and I don't see any harm in that. And if somebody makes a few bucks for their extra effort, fine by me. So I'd encourage you to have a look at some of these. They're all short, they're all skill-based, and they're as focused as we can make them, so we're not wasting your time. We have some traditions here. I've been here for 30 some odd years, 33, I think. And so we've learned a few things that work and a few things that don't. And I like to have some fun at work, and bees are, they're such a fascinating insect to work with. There's lots of things we can do for fun. So this was our tradition here. Is the first swarm of the year, our newest employee gets the opportunity to catch the swarm. We try not to have swarms, and so some years we don't even get this opportunity. But this was Janine, and this was the biggest swarm I have ever seen. There were 8 queens in that swarm. And so I thought, what a fantastic opportunity. Like, it's low to the ground. We've got this really passionate new beekeeper that wants to learn things. And so I thought, before you do anything, Janine, pretend you're going to give that swarm a kiss, and it'll make a great picture. Well, was that such a good idea? Yeah, maybe not. Well, poor Janine, looking pretty sad there. But that was a number of years ago, and she is still a beekeeper, so everything worked out in the end. I'm going to talk a bit about some of the research that we do here. Again, our core mandate is research and teaching. So virtually all the work we do is based on honeybee health. And as you know, with all the issues that bees have, we've got a lot of work to do. Back in 2007, when we started experiencing much higher colony losses, we really wanted to just stand back, take a look at what was going on, and try to figure out what's new, what's different, why are the losses higher. And so we did a study that lasted 2 years, and we tracked 413 colonies in different regions of the province, and we measured everything we could about all 413 colonies over a year and a half time frame. In the end, we looked at the colony mortality of all these colonies and then correlated that to the factors that we had measured, so we could determine what were the most significant factors associated with colony mortality. No big surprise, Varroa was number 1. It still is number 1. Nosema was number 2. Environmental factors were number 3. And hive management was number 4. And of course, all of those bear more explanation. I don't think we'll get into that too far in this talk, but we do, with this project, we collaborated with the tech transfer program to relay what we had learned, and it's set a focus for their activities and a focus for our activities ever since 2008. It kind of laid out a framework of what we should be researching, what was important, what was less important, so we could focus on where we could make the most significant impact. Backing up a little bit, when I first started here in 1987, what we were working on was tracheomites. They were new to North America. They were causing significant problems in the United States. We didn't have them here yet, so we were going down into New York State to study them. So we're going back and forth a lot into the US to study these tracheomites. From that, we developed a breeding program where we could breed for resistance to tracheomites. We were fortunate. There is enough resistance in the bee population that over time that this resistance would have happened naturally, but we were able to accelerate that resistance by breeding. So tracheomites are no longer an issue at all. You rarely hear about them, but we're able to speed things up a little bit by the breeding work that we did that way. Just a few random notes here about the research that we're doing here. This is an example of, like, we have to come up with techniques to study things. And very often we're studying individual bees. So this is an example where we're studying 8 different stressors that affect bees at the individual level. So each of those paint marked cells contain a larva that was fed, now this is kind of a hard thing to explain to people, but we gave each of these larvae a sublethal dose of an agricultural insecticide. Not enough to kill them, but what they would be exposed to in the environment. And then we looked at them throughout their entire lifespan to learn what these sublethal amounts of insecticides do to bees. Here those bees are later in their life, where they're paint marked as adults. In this particular project, we had marked 43,000 bees. That took 8 people 16 days to mark them and treat them, or to mark them at this stage here. They were treated earlier on. And then we monitored them throughout the rest of their life by observing them in observation hives and nucleus colonies with entrances that were extended, so we could see them going in and out and monitor their activity. So bee research, there's a lot of tedious work. We've got a lot of students around here, a lot of volunteers, and so we kind of wear them out with some of the tedious work. That's maybe not the right way to put it. So, of course, Varroa destructor is our #1 problem, and what we're focused on most. For those of you that maybe are a little bit newer to bees, you can see the Varroa mites here on the pupa on the left of your screen. So they feed on immature bees, but they also feed on mature bees, as you can see on the bottom right. They're incredibly well adapted to life on a honeybee. The top right, you can see a Varroa mite that looks kind of like a turtle in a shell, but they're domed and they're stuck right down on the bee, and it's really hard for a bee to even pry them off. The one on the right is flipped over. You can see the legs there. If you count them, there's 8. They're somewhat related to spiders. They're not an insect, but their mites are in that category, arachnids. So what do we do about Varroa mite? We've got a multi-pronged approach to dealing with them. One of them is testing materials that work as miticides. We've done a lot of work with different essential oils, one of which is thymol, but we've also used oregano, clove oil, a few different things, and we've come up with good methods of delivering thymol and oregano, very cost-effective methods. This is thymol mixed with icing sugar, put on a sheet of newspaper and set on the hive. It's that simple. 95% effective against Varroa mites. We've tested this time and time again in different jurisdictions, but we're having a real problem with getting this product registered. No major company is interested in taking this on because it's too simple, it's not patentable, and it uses a natural material, which, so we're struggling. We're trying to get this product registered. Since then, we've come up with a method of delivering oregano. We mix the oregano, which is from plant oregano, with glycerin, and then we soak it in a pad and put a shop towel, actually, and put it on top of the colony. 90% effective with that one. But we're hoping, we keep putting it out there, we're hoping that this gets picked up somehow and we're able to get the product registered so you can be using them in your colonies. In the meantime, there is a thymol product that is registered, not quite 95% efficacy, but it is another natural product that can be used. Please don't experiment with these products at home. They need to be delivered at the right dose, in the right amount, and in the right way for them to be effective. So here we are using oxalic acid. We did not invent this method. It was developed elsewhere, but the Canadian Honey Council pushed through the registration of this product. So it's a very cheap, very simple product. We mix it in with sugar syrup and deliver it to the colony late in the fall. It works well as a good backup treatment for mites, but late in the fall is too late if you have a significant mite problem. How we use this product is as an alternative, so we're not just using one product time and again, but we switch in the oxalic acid late fall to kill off any mites that might be developing resistance to other products they'll be using. So we're also breeding for resistance to varroa mite. You can see that LVG in there. I can talk to you another time about that project. It's a pretty involved project, but LVG stands for low varroa growth. We've determined over time that grooming behavior is the most significant behavior that bees exhibit that gives them resistance to varroa mites. The ability of one mite to take, the ability of one bee to take the mite off its own body. To test this, we put a bee in a petri dish, we put a mite on its back, we cover it up with saran wrap, it's very high tech, and then we use 2 counters and we count the bee's performance in trying to remove that mite from its body. So it's much like scoring a boxing match. If the bee is really good and very aggressive at getting that mite off its body, it gets a high score. We've determined that colonies that exhibit this good grooming behavior also have a low varroa growth rate. So to measure the low varroa growth rate, we measure the mites that drop onto sticky papers in the spring. We measure the mites that drop onto sticky papers in the early fall. And the difference between those 2 measurements is the growth rate of the colonies. We can then treat our colonies in the fall. So this is not a survival method of breeding bees. I don't agree with that method at all. I don't have the conscience to let bees die to be breeding them. So we treat the colonies in the fall, but in the meantime, we've learned which colonies exhibit low varroa growth rate, and we can use breeder queens from those. What we've done in this project is we've selected for high growth rate, we've selected for low growth rate, and you might wonder why we would look for high growth rate. But what we've done that for is to be able to learn fairly quickly whether what we're doing is working, to see if there's a separation between those 2. And we found that yes, we are gaining success with that. The colonies with the low varroa growth rate also have low viral loads and even low nosema loads. So we know this is the future. And so we've created 4 new videos on this to be showing to queen breeders so they can incorporate this method, a very simple method. Anybody can do it, but you need to have a large number of colonies to be able to select from, and you have to commit to it. But any of the queen breeders in the province that are committed to breeding varroa resistant stock will be adopting this method. We're also working on nosema. It's number 2 or 3 of our problems. It's a pretty cool little organism, despite the fact that it affects bees. But you can see the one little nosema spore there. And then on the left, you can see the nosema spore that has germinated. So that long tube you can see coming out of the spore, that's what happens inside a bee's gut. So when that spore gets in a bee's gut, and it has to be a honeybee, like a very specific, the spore germinates and it sends out that long tube, which punctures the wall of the gut, a cell in the bee's gut. And then all the genetic material moves through that long tube into the honeybee gut cell, and then it reproduces in there. And then when that cell is totally full of nosema, it ruptures, the bees defecate the spores out, and they can spread from bee to bee in that way. So what we're looking at now is alternative treatments. We're looking at prebiotics and probiotics. We've gone from lab trials with hundreds of different products to small-scale field trials to large field trials. And we now are quite confident that the products that we've chosen are very effective. And some of these are also used in chickens, pigs, and other forms of livestock. So we hope that the registration for these products will be a little bit easier than the miticide. Other research topics, just to pick a few here. Agricultural chemicals, did a lot of work on neonicotinoids. My emphasis on that was trying to find out what's coming into the colonies and what quantities and what effects that's having on mite or on bees. We're also looking at miticide toxicity to bees. So some of the miticides that we use can be toxic to bees. Formic acid is one that really whacks bees, but it also works for mites. So it's a trade-off there. But we found that the Apivar, the product that is the synthetic product that's currently registered, has very low toxicity to bees. So it's quite a safe product to use, more so than any of the other synthetic products that we've tested. Our thymol product also has extremely low toxicity to bees. We're looking at heritability of different traits of bees, biological control of mites using fungus and other things. We're the first in the world to figure out how to cryopreserve or freeze bee semen. So we're able to keep bee semen viable over time. We work together with people in our animal science department on that. And honeybees are one of the most challenging things the semen to freeze. Elephant semen is about this long. I don't know if you can see my fingers here. Drone semen is about this long. They have very, very long tails. And when you freeze them, they get all tangled up together and break. So what we do is we dilute them so they're all spread out, and we add cryopreservation agents, and we're able to keep them alive that way. So they could be used for insemination in the future. So there's other people that have picked up on that and are taking it further. We haven't done much with that lately. We've also researched organic methods of managing pollen. A personal interest of mine is what bees are working on. I have a presentation on that I can show you sometime. But I'd just like to point out bird's foot trefoil. Have a look for bird's foot trefoil. If you don't know what that is, it's a low-growing yellow flower. You'll see it a lot on roadsides, and it blooms for a very long time frame, from June until September, and it is a major source of nectar that's widely unrecognized. But we send samples off on a regular basis to a woman named Melissa Girard in Quebec, and she analyzes the samples of honey, centrifuges them to extract what little amount of pollen is present in the honey, and then identifies them by recognizing what these pollen grains look like, figures out a percentage of what's in the honey, and then we get these results back from here. So you can see a few of the other major crop plants that bees are working on from looking at that slide. Lots more I'd love to talk to you about on that subject. There's Melissa Girard's contact information. It's also on our website. We have a whole section on plants for bees that I encourage you to have a look at. And Melissa's contact information is on there as well. Just a little point of interest, the sample on the left and the sample on the right were collected from our pollen traps on the very same day from two hives that were sitting side by side. So you can see how much of a difference there is from hive to hive on what bees are foraging on. And that's a good demonstration of how the bee dances really work to get bees going where their nestmates are telling them to go. Just going to mention a few of our beekeeping methods that we use here, that some of which might be adaptable for you. We use bee escapes for harvesting our honey almost exclusively. So here we're putting bee escapes on colonies. We use a canvas inner cover. This is the #1 question we get from our YouTube series. I've answered this question over 1,000 times, despite the fact that we have a frequently asked question list, but at least every day I get somebody ask me, what do you use for inner covers? It's 18-ounce canvas. And if you look at our frequently asked questions on our website, you'll find sources for that. I really like it because you can it's cheap, inexpensive, light, easy to make because you just cut it to size. And you can peel the inner cover back and have a look inside. There's no scraping the top bars because they don't put wax there. And it saves us a lot of time. It's quite handy to use. We use pig ear tags for giving our colonies numbers and standard label maker to indicate what queen is in the hive. We use a clipboard cell builder method when we're raising queen bees. And we have several videos about that if anybody's interested in getting into queen rearing. Here we are with a group of students helping out with preparing these cell builders. So we shake every bee off every frame in the cell builder to check for any cells that the queens might be raising themselves on their comb. If they happen to raise a queen on their own and she emerges, it will destroy all the cells that we've been trying to raise ourselves. So we have to shake every bee off, make sure none of those cells are there. The students on the right are new employees. So they check first, and the fellow sitting down there, Dave, is a longtime employee. And so he double checks the frames. And any of the new students that miss one cell are fired on the spot. We hire them back again in another 5 or 10 seconds, but we have a little drama, and a little fun over that. And people know that this is really serious. They have to find every single one. Once those cells are 10 days old, we put them in a $1,000 incubator you can see on the left. And we keep them at 34C until we get those queen cells into the nucleus colonies or the colonies that they're going into. But on your right, you can see a $20 device that any of you that want to handle queen cells can purchase. It's a small cooler with a rubber hot water bottle, 4 inches of shavings. You fill that with, you run your tap until it's as hot as it gets. Fill up the hot water bottle, put 4 inches of shavings on there, nestle your queen cells into that, and you have 5 hours to get them into a colony. So it's very simple to do, and we've done a number of tests on this to show how effective this is. I really encourage people to use queen cells. I think they're in some ways better than mated queens, but there's lots more we could talk about there. But if you find you're looking for queens in the spring to make up splits, first of all, super your hives when they need to. You don't have to make splits first thing in the year. But if you're able to buy mated queens, great. You can't get them until the middle of June if they're from Ontario. But you can get queen cells a little bit earlier. And we encourage you to consider thinking about selling them. I sell nucleus colonies from my own hives. And at the same time that people come to pick up nucleus colonies, I have queen cells available. So they'll pick up a nuke. They'll pick up 2 or 3 queen cells and they'll go home and they'll requeen their other colonies or make up a split and put in a queen cell. So it works for me, it works for them. We use a variety of mating nucleus colonies, and we have to put bees in them. So this is showing collecting bees. We shake them through. You see I'm holding onto a frame there, and what we do is that there's actually a funnel built into that box, the vertical box you see there. So we just bang that frame down, the bees drop into the screen box, they have frames of honey on there to cluster on, and that's how we're able to capture bees from brood chambers and move them into our nucleus colonies. I've already mentioned our isolated mating yards and enough about drones, but this is our island in Lake Simcoe where we're setting out little styrofoam mating nukes. We've given up on those on the island. The coons have learned to destroy these nukes to get the syrup that could be in them or to eat the comb. And it was an arms race with the coons and I lost. Coons won. So we're now gone to wooden nucleus colonies. But for whatever reason, coons in our local area here haven't figured this out, and we're able to use these styrofoam nukes here. So this is what we've gone to using on the island. It's a 4-way mating nucleus colony. So it's a medium depth box divided by 4. If we look down on from the top, as you can see on the right, there's a partition across the middle, there's a partition side to side. And so there are four nucleus colonies in there. And on your left, you can see a queen cell that has emerged in one of these little nucleus colonies. I really like these nucleus colonies. And if anybody's interested, you can ask me more about that. We've been working quite a bit on banking queens through the winter. So this is a picture showing a block of queens that we're putting into one colony. So we remove the queen from the colony. We put a number of queens, this case 24, into that colony and bank those queens through the winter. We're up to about 80% survival of these queens, but it's an ongoing project that we're working on incrementally improving. We're getting our best success now with banking these queens indoors in colonies that are kept at 15 degrees C, so quite warm. But by doing that, bees never cluster, they can move around, and so they never actually leave these queens and take good care of them. So we've got more work to do on that, but this is a great way of getting queens that are produced at the time of year when nobody wants to buy them, i.e. August, into the spring where everybody wants to buy them and they can be available. This is an outdoor method of banking these queens. We super insulated them, again, to help prevent that clustering. And these are like super strong hives wrapped up in a way that they do in Western Canada with their colonies. So they're super well insulated. And we were getting about 60% outside, but this method indoors is working better for us. And we were working at this, chipping away at it on our own. And eventually I realized that a lot of beekeepers have great ideas. We need to get other beekeepers working on this. So Les Eccles with the Tech Transfer Program has helped with that. We've got a number of people working on this, and some of the beekeepers have come up with some great ways. And so we're learning a lot more by kind of crowdsourcing the research. So now I'm going to go through a few quick points, some of the tips, tricks, and tools that we use here. Whenever we see a queen, we pick up and cage her. I use these little, they're made by a company called Nyclo, these cages that are open up easily. We can pop a queen in there. Our new employees learn how to pick up queens the first week of work, and it takes a bit of practice, but if you want to try this, practice on drones. They're a little less valuable than your queens, and you can learn how to handle these. Just a little anecdote there. Look at that bee in the middle bring propolis home. You see that propolis on her legs? Isn't that a beautiful color? So she's bringing propolis home, and then, the one thing I don't understand is how they get it off their hairy legs. I can't even get it off my fingers. How do they get it off their hairy legs? So maybe one of you guys can answer that question. One of the things we train brand new beekeepers, their first day of taking a course, is how to see eggs in the colony, because it's a really useful thing to be able to diagnose what's going on in the colonies. So the first thing we teach them is how to pet bees. So you guys pet your bees? Anybody pet your bees there? What we do is just lay our hand on the bees. Look at your hand nice and flat, and we'll lay it on the bees. And what happens is they move out of the way, and you can see what's underneath those bees. And you're much more able to see larvae, pupa, everything like that underneath, and especially see the eggs. This is some black plastic foundation, and so the eggs show up really well. I hope you can all see that at home, but there's eggs, and there's tiny larvae that are in the middle of the picture. There are the age that we would use to graft larvae for queenery. But to see them, clear the bees out of the way, hold the frame so the cells are pointing straight towards the sun, and you're in the middle between the sun and the frame. So your eyes are looking in exactly the same direction. The sun is shining into the cell. Tip the frame a little bit this way, a little bit that way, and all of a sudden you'll say, aha, I see the X. We use single brood chamber colonies here. This is something that was very rare 30 years ago, a little less rare 20 years ago, and now over 60% of the colonies in Ontario are managed in a single brood chamber. Almost all commercial beekeepers manage their hives this way. So it's something we started out with 25 years ago and have been trying to promote that. But we find there's a lot of advantages of managing colonies that way. Last picture, I'm going to just point out a few things. This is in one of my own bee yards, but the colonies you see here are double nucleus colonies. So that bottom brood chamber actually has two hives in it. There's a queen on the left, there's a queen on the right, and there's a partition in the middle. I sell a nuke out of the left, I sell a nuke out of the right. Those are $220 each. So by the middle or late May, I've got $440 of value out of a colony. And then I can go on to produce a honey crop from them. Put a queen cell in each side after taking out a nuke. Keep those two colonies separated by putting canvas over so that there's no way the queens can get back and forth over the partition. And then fold that canvas up once the queens are mated. Put a queen excluder on, add the supers, and then the bees share the same supers. So the bees on the right go straight up, the bees on the left go straight up, and they produce a kind of honey crop together. And that way I'm able to winter these colonies, harvest the honey, and winter them as two separate units to go on to sell nukes from next year. In the background, you can see a white jug on my truck. We have water on the trucks. I may be in the wrong profession, but I hate sticky hands. So I've always got water handy to be able to wash my hands. And if I'm in somebody else's truck, I generally have a drink of water or a water bottle with me. So I'd like to demonstrate to you how to do what we call the Monty spit wash. It was taught to me by Paul Montauk. Gord probably remembers Paul Montauk. He passed away about 12 years ago. He had a lot of great ideas. Most of them were pretty rough, but they're great ideas. So what he did to wash his hands, he'd take a drink of water, fill up his mouth full, and then squirt a nice steady stream out of his mouth onto his hands and wash up his hands. And it was way better than trying to pour water on your hands. So every time I wash my hands that way, I think of my buddy Monty. We use flatbed wheelbarrows here, the two-wheel kind. You can get these in Canadian Tire on sale. And every year or two, they come on sale, a two-wheel wheelbarrow. They get rid of the steel bucket and replace it with a flatbed with a rack at the front there. And that's 300 pounds on that flatbed. And I'm holding on to it with one hand. So it's a really great way of moving heavy weights around to and from the beehives. Sometimes it's simpler to use that than one of our trucks to get things moved around. There's pictures of, I don't have exact plans for that, although we have plans for some other things on the website, but there are some pictures of construction methods for that on our website. I make and sell tool belts for beekeepers, a little bit of shameless self-promotion, but Dancing Bee, Innisfil Creek, many of the other bee suppliers sell these belts for me. And they mean you never lose your hive tool. You don't never have to worry about where you set it down. You don't find them with your lawnmower as you're cutting grass. And we can keep a few other tools that we want to carry on from place to place that way. But there's a magnet there that holds onto the belt. And so you no longer have sticky pockets and so on. All right, I'm done my self-promotion. But every bee belt comes with a free queen cage. This is something I think every beekeeper should have. These little aluminum stands. Again, you can get them at Canadian Tire. They come on sale periodically, but they weigh like less than 5 pounds. and they'll hold 300 pounds. And so you can see it's set off to the side of the colony, and I just swung all the supers over there at a comfortable height, swing them back into place after I put the bee escape on. In the fall of the year, to harvest the honey, I leave the supers stuck together, and I just tip them back onto that stand, put a bee escape on, and tip them forward. No lifting at all. And for other, you can use them for a multitude of other purposes as well. So they're a great little tool for beekeepers to have. That's it. I'm done. Thanks for your attention, and it's been nice to see a few faces there and being able to see a little bit of response there. I'd love to come up and see you when we can talk in person. Oh, I guess I could mention we've got a $12 million project underway with building a new center. We started off with much more modest goals than that, but we found a donor that wants to pay for half the building. So she's in for $6 million donation. Not bad, huh? So we really only need to find one other person to get the other $6 million. Ivana, you smiled there when I said that, so maybe you're thinking you might want to help us out with another $6 million. But what the heck, huh? No, we're reaching out really broadly, or we're getting going, but we've been struggling a little bit with this project because it keeps changing. We found one site that turns out that site didn't work that well, and so we moved to another site. Our donor really wanted a spectacular building, and to do that, we had a design competition. The first time the university had ever done that, but that was thought to be the best way to bring in the best architects. And so we did that. That was a big project. And we now have the firm Moriyama and Toshima. They designed the Ontario Science Centre, the Canadian War Museum, and many other iconic public facilities. So they're going to be working on this project, and an emphasis that we're building is our public outreach with the new center. So we'll be able to do much more education for beekeepers and for the general public in the new facility. It'll be set up like a visitor center where anybody can come and do a self-guided tour of the site. And we'll have guided tours as well, discovery center inside, and much better teaching and research facilities, and our production facilities as well. So it's really going to be the only center in the world that will bring all those activities together in one site. And we're going to try to build this to net zero for energy consumption. And it'll be an example in many different ways of architectural design and sustainability. And that also includes the grounds of pollinator gardens and research apiaries and demonstration apiaries. So it's a huge project. When I started into this, I thought it might take a couple of years. We're 4 years into it now. And I talked to Marla Spivak in Minnesota, and they built a new center there. It's not quite as ambitious as our project, but she said it took them 50 years start to finish on that project. So
Did you say 15 or 50?
15, 1-5.
Okay.
Even that was
I was going to
demoralizing.
say she's not that old.
No, but so it's just a long process, and at the moment, our biggest drawback is I can't show you pictures. I can show you a whole bunch of pictures of what we're not going to build, but that's not going to help us out too much at this point. So we're hoping soon to have some images that we can share of what we're going to build. So stay tuned. It'll be a great place for you to come to visit. We hope to have, we're reaching out really broadly, hoping that we can get you know, $10 here, $10 there, $1,000,000 here, $1,000,000 there, whatever people can afford or what they feel like they want to support with, we'll welcome any amount. So we're already set up with that on our website. We have a donate button, and but you'll be hearing more from us on this project as we go along.
Paul,
there was,
there's a question on your chat, Paul.
Okay, just check that out here. I'm looking for that chat.
It's on the bottom.
Michael, you'll be able to help me.
basically it was, how do you know there's more than one queen in a swarm and how do you separate them out?
Well, thanks, Bert. That helps. What we like to do when we're catching a swarm is dump the bees on the ground in front of the box they're going to be going into. Rather than capturing them in the box, we dump them on the ground and let them run in. And when we did that, you can often, the queens will end up on top of the bees, running across their shoulders. It's kind of like crowd surfing for bees. You get the queen kind of motoring in there, and it's often possible to see the queens coming in, and we counted 8 in that one going in there. What happened there is I dropped the ball, and we had a bunch of these double nucleus colonies that got far stronger, far quicker than I anticipated. A lot of them were preparing to swarm. We had a period of rainy or cool weather, which prevented any swarming from happening. And then we had a nice sunny day, and all this pent-up swarming happened all at once. And so all these bees were in the air, and they just merged together, which can happen. And so that's what happened there. So it was not a good thing, but we at least got a few nice pictures out of it. Oh, here, I got the chat here now. I don't see any more questions.
With the good weather we've been having and with the increased, seems to me, increased brood in the hives now compared to perhaps other years, is swarming going to be a bigger concern, do you think, this spring and maybe a bit earlier?
Well, that remains to be seen. We've had 3 crappy springs in a row, and I sure hope this one changes that pattern. Even last year we had, really nice temperatures in March and early April, and then it got cold, and it stayed cold till the end of May, and so that suppressed colony growth and delayed some of our beekeeping activities, queen rearing, splits, and so on. And so if the weather continues to be good, that'll be great, but we will have to watch for swarming. As you know, you have swarm years and you have years that are just as hard as any swarming at all. So keep an eye on that. If it does carry on to be as nice as it is, I'm hoping it does, but we'll have to be thinking about preventing swarm and controlling for swarming. And I divide those into 2 things, but swarm prevention is supering in a timely manner, keeping younger queens in your colonies, having the genetics that have a low propensity to swarm. Our buckfast bees have that. And making up splits, doing some other management things that help to prevent that swarming. Swarm control is when you actually have to get in and remove queen cells to stop them from swarming, which we have a video on that subject. It's not, it's doable, but there's a fair bit involved in doing that. So the best thing is to do everything you can to prevent that swarming from happening. And one of the most significant ways is by giving bees enough space. And don't think of it in terms, like when you're adding supers, you maybe aren't thinking of it in terms of when the bees need the supers for the honey. Think about when the bees need space for bees. and you might be supering a little earlier this year than normal. Some strong colonies, like we use deep root chambers as single, and we use medium depth boxes. But some of, like if we have a really strong colony, we might add 2 honey supers to that even as early as the beginning of May. But every year is a bit different. You have to watch your bees, and they'll tell you what you need to do.
Well, there's been a number of questions going back and forth, and Adam's been good enough to answer some of them, but just in case somebody's not seeing their chat, it's relating to pollen. Tom had a question earlier about white pollens coming into his hives. He wondered what that was from. It may have been answered, but
Don't know. White pollen. Sawdust is pretty light in color. Bees do collect some of that pretty early on. Right now, they're switching from silver maple to willow in our area. So silver maple is kind of a greeny beige, and red maple the same. But willow is a kind of a bright yellow. So we're starting to see more of that coming in. We'll see a little bit of very dark looking pollen from the plant Scilla that grows in a lot of people's yards. That's abundant enough that you see some of that. A little earlier, we were seeing pollen from snowdrops and crocus. It's more orange. So if you see a bee on a flower and it has pollen on its legs, honeybees only forage on one floral species on one flight. So you can really learn what color the pollen from a specific flower is by seeing what pollen the bees are carrying when they're working on that flower. And then when you're back at the hive, you can recognize what's coming in. And I have a lot of fun trying to learn that. I'll never finish learning that, but and I forget a little bit from year to year, and have to learn it over again every year. But what's white, I don't know. You might have something different in your area.
The other thing too is the bees are raiding my bird feeders big time. They'll empty them out in half a day, digging them out. And someone said maybe they're after the corn dust.
Yeah, they pick up that chaff there. It might have a minor amount of nutritional value. I have one bee yard, and every fall, after there's not much blooming, if we get a nice day in November, my bees in that particular yard are bringing tons of pollen in there, what appears to be pollen. And then same thing in that yard in the spring. And what they're doing is they're going to a neighboring dairy barn, and they're getting the chaff out of the feed that the cattle are being fed. Very much like your bird feeder there. So there may, in that case, be a minor amount of nutrition available. But I was out cutting with a chainsaw this summer, and when I set my saw down, I had bees all over it, collecting sawdust off the chainsaw, which of course smelled like chain oil and exhaust and gasoline and everything else. So it's amazing what they'll work on. Ducks and chicken mash. Oh, yeah, until there's some real pollen coming in, they'll take what they can get. And one year I had bees collecting rubber dust from a pile of burnt tires. And they were rubbing all over these burnt tires, collecting this black dust, carrying it back home. And then they were using it like propolis too, because they smeared it all over the inside of the hive. The hive smelled like rubber. It was black. There's 2 hives in that yard that did that. I couldn't even harvest the honey for those colonies that year. So it's kind of shocking what they will work on. You think bees are smart, but sometimes they make poor decisions. So let's put it that way.
they're being trained to work with drones to spot mines or to detect mines.
Yeah, the
Any
landmines,
personnel
yeah.
mines, apparently.
Yeah, yeah, and it's amazing what, you know, and I gather they're smelling the explosives in that case. They're trained to recognize that. But, yeah, and also in, we do some of our research involves looking at proboscis extension reflex. So that's, we call it PER because that's hard to say, proboscis extension reflex. So what you do is you hold a bee in something that's very much like a hollow Bic pen, and then you have their head projecting from that. It sounds pretty terrible, but then we train them with a scent. So we blow a scent over the bee, and then we feed the bee a drop of syrup. And we would keep repeating that process so that they learn to extend their tongue whenever they smell that smell. And then we can use that learning behavior. And again, it's kind of hard to talk about, but we treat these with agricultural insecticides or bromites or other things. And we see how that affects their learning behavior using this proboscis extension reflex. And that's a technique that's has been used with some success for detection of drugs in airports and things like that. I don't believe any of this has got to a commercial level, but you know, it's possible that that can be done. Rust fungal spores. That's interesting. And that's very true about the sugar concentration, how they can figure that out, what plants are producing the best concentration of sugar. Yeah, bees are pretty amazing. The whole, this sawdust and other things, it's a bit of a surprise. But. Okay, were there any other comments or questions?
Oh, I have a question regarding spring feeding, sugar syrup. I just did a full inspection of my hives today, and I found that I had at least 2 full frames of honey left in each of those hives. Since I've got that much in there, do I need to be considering giving them sugar syrup?
I would hold off for now. You might want to be doing that a little bit later. Keep a good eye on it.
Okay.
If we get, right now they'll have started to build up brood. So they've cranked up thermostat. So they're going through a lot more feed now than they were earlier.
Mm-hmm.
And if we get into cold weather, that'll be even more so. They'll eat even more so. We generally don't feed in the springtime. The odd hive, it's a bit light. I'll stick a frame of honey in from another colony after inspecting for foul brood, and that'll get them through. Last year, we had, as I mentioned, some good weather earlier on, but I looked at the long-range forecast at one point. The hives were getting pretty light, and there was going to be 2 weeks of cold weather. I ran around, and I got feed on all our hives just at the last minute, and that saved a whole bunch of beehives because we had that cold weather coming. So keep a good eye on it. You may want to be using feeding a bit of sugar syrup a little bit later. Once we get into the dandelion flow, as long as we're getting, you know, periodically some good weather, like that flow starts and stops depending on the temperature. But once we get into that, usually the bees are going to be okay on their own without feeding. But every year is a little bit different, so just keep a good eye on it.
Okay, that's great.
Any other questions for Paul?
Well, it's Dale still. I'll throw out another question I've got here. Water source. I don't have a local water source for my bees. The closest natural water, I would say, is about 300 meters from my yard. And I was wondering about trying to get something close by. I've put a bird feeder, or sorry, a bird bath oh, it's probably 50 to 100 feet away from the yard. And the bees are not at all interested in that. So it tells me they're getting water somewhere else. But do you think I need to be concerned about that?
It's Dale, right?
Yes.
Yeah, thanks, Dale, for a great question, and I'm afraid I don't have a great answer. They kind of do what they want to do when it comes to collecting water. It is possible to get water out for bees, and that is a little easier to do in areas where it's very, very dry and there's not much available. But they kind of get trained onto certain water sources, and it's kind of like when they're bringing nectar home from a flower, there's a scent to the nectar. Well, there's a scent to the water as well. And so bees learn to recognize that scent, and then they orient to that as they're flying out to find water. So for example, swimming pools that have chlorine, and the chlorine itself acts as an attractant to bees. So once they're onto a swimming pool, it's really hard to get them off that. I have one pool that's near, one bee yard that's near somebody that has a swimming pool. I put out a kiddie's pool, I put water in it, I make a little beach for the bees so they can just walk along the beach and suck up the water. And the bees fly right over that and go to the swimming pool. But at least I can tell the landowner I'm doing everything I can.
Yeah. Okay.
I don't have a great answer there. One thing that other beekeepers have told me is that they suggest, or if they have a pool themselves, they'll hang a towel in one corner of the pool. So it's into the water and it's soaking up the water. And all the bees go to that spot because that's a great spot for them to get the water. And then they're not in the other areas. Nobody likes it when I tell them that answer. It really is only a good answer for a beekeeper with a pool. But some other people have had success with using very shallow pools of fresh water, and that I think is key. Once it gets stagnant, the bees are really not so interested in it, and that's the problem with my kiddie pool. But if you have a very shallow pool full of pebbles or something that the bees can walk on, if you watch how they collect water in nature, they'll get to the edge of a stream or pond or something, just where the soil is wet, and then they sop it up or stick their tongue into the edge of the water and drink that. But they need something to land on and to be able to sop that up. So you can experiment with that and let me know what works, okay? I need some answers myself.
Okay, thanks, Paul.
You're welcome, Dale.
Okay, well, it's going on 9 o'clock and Paul's been talking quite a bit. So unless there's more questions, I want to say thanks very much, Paul. Certainly enjoyed your talk as always. And again, I always learn something from it and from the rest of the people that are participating as well. So thanks very much.
You're very welcome, Gord. Thanks a lot for inviting me to speak to your group. And I'm happy to still see you're still leading your flock there. Or your swarm, I guess would be a better way of putting it.
Not very effectively during this time, but yeah.
All right, well, keep it going.
This works.
Things will get better.
All right, well, thanks again, Paul. Appreciate that.
My pleasure. Thanks, Gord.
Thank you.
Thanks, everybody.
Thank
Nice to see
Thanks,
you.
Paul.
Good night.
Okay, good night.